And we acknowledge how very busy you all are, and we really, really appreciate you being here today. As Ashraf mentioned, today's topic is crucially important given the gravity of the pandemic, and will provide an overview of the vaccines in the workplace, some of the science around the vaccines and the implications of the 501YV2 variant. And even more important from an occupational health perspective are the programmatic issues that need to be considered in a vaccine rollout. It is very encouraging that thousands of our health workers have received their vaccine shots in recent weeks, and many more to still follow from other categories of workers. Colleagues, we trust that this session will be informative and beneficial to all, and that it invigorates pro-vaccine activism among your networks. I would like to once again thank all the presenters today for your time and your willingness to share your expertise and experiences on the great work you are leading, and the delegates for making the time in your busy schedule, as well as the NIH team for organizing this session. May you continue to be ambassadors and advocates of health and safety, not just for COVID, but for all other hazards in the workplace. Colleagues, I don't want to take up much of your time and instead spare the minutes for the presentations and the Q&A instead. May you have a very productive session today, and do take care. Back to you, Ashraf.
Thank you very much. That was Dr. Tanusha Singh, the chairperson of the NIH's COVID-19 Occupational Health Outbreak Response Team, the OHOT team, as well as head of the Immunology and Microbiology section. Thank you very much for that introduction. And without much ado, a little bit of admin, just to remind everybody that the hands raising function as participants and as attendees, your hand raising functions, your microphone, your video functions, and so on have unfortunately had to be deactivated, and only those of our presenters and panelists who's going to answer your questions in the Q&A box have these promoted functions. Please use the Q&A box for your questions related to this particular content of presenters as they come up in the sequence, and only the chat box for general administrative questions like Msibi's hi that I just see now in the open section of the Q&A. So Ms. Mombusa, please, if you could just put your general comments in the chat box. So I don't want to delay anymore. The issue of attendance and CPD certificates have been covered in the slides that have gone on, and I will post in a second the YouTube link for you in the chat box to share with your colleagues when we reach the maximum limit. So our first, the program as we've have it, the topics for us today is in the following sequence: COVID-19 vaccine science, development and safety by Dr. Simba Tavuka. The second topic is the COVID-19 vaccine workplace program considerations, and that's Dr. Azwi Takalani. She will be presenting on that one. The third one is COVID-19 vaccine workplace, COVID-19 vaccine for workers, occupational health perspective by Dr. Sipho Senabe. And then we have the fourth topic on occupational health-based clinical trial in vaccination and occupational medical practitioners perspective by Dr. Lumka Puwani. And then the final topic is on vaccines and SARS-CoV-2 key take-home messages by Prof. Elizabeth Maine, and we are very thankful, as Tanusha has indicated, for you to dedicate your time to us today. So without much ado, if I could then just quickly, briefly ask Dr. Tavuka to share the screen and to check the audio. And by way of introduction, our first speaker, as I mentioned, is Dr. Simba Tavuka. Tavuka. The Dr. Tavuka has an MBCHB, MSc, a DTM&H, a medical epidemiologist and lead safety physician medical monitor in the important Sisonke open label trial. I immediately hand over to you, Dr. Tavuka.
Good morning. Thank you to the NIOH for inviting myself, Simba Tavuka, it's Takuva, and my colleague, Azwi Takalani, to come spend the morning with you and discuss COVID vaccines. So my role will be to give a really general overview of COVID vaccines, and I'm happy that there are experts in the room that will get into more depth. I saw we've got Prof Elizabeth Maine as well, yes. So the presentation is pitched so that one does not need a deep understanding of immunology or vaccinology to understand. It is really gentle. I am also not an expert in virology or immunology, but as you've just said, an epidemiologist working in clinical trials. So I hope it will be very understandable, and we really look forward to your questions as well. So as an outline, we're generally going to look at how vaccines work, how they are developed, and also look at current vaccines in the global market. Perhaps before we start, I would just like us to take a step back and look at the sobering schema on the slide. So this is as of February 9, 2021, and it shows the daily COVID-19 vaccine doses that had been administered. And globally to date, there are close to 200 million doses that have been administered. And when you look at the distribution on that map, it's quite sobering and it's quite worrying, the pattern that is actually displayed there. However, it is great to note that there are several mechanisms, including the COVAX facility for the African Union, that have been set up to support and hopefully speed up access. As of that time, the countries that have really started maybe rollout might not be the appropriate term, but administering vaccines through either a rollout or a trial or implementation study or any form, they're really few in Africa. And as you can see, you've got South Africa there, you've got Zimbabwe, Egypt, Morocco, Algeria, Rwanda, the Seychelles. And the past few weeks, the vaccines have started trickling in to the other countries. For instance, Ghana, we're reporting that they've received COVAX vaccines and we're about to embark on a vaccination program. But it's still really worrying, the speed with which we've caught up as a continent to vaccination. So, what could a vaccine in general do? If a vaccine was safe and efficacious, what exactly would you require it to do? So when you look at this, it's always best described at two levels. So first you look at the benefits of a vaccine at an individual level, and then look at a community-wide benefit level. So looking at the individual, ideally your great vaccine, you would want it to be able to prevent infection, to protect individuals from being infected with disease. And then also, you'd also like it to be able to reduce the severity of illness, such that even if you are infected with the virus or germ, the severity of illness is quite reduced. So that's looking at it at an individual level. Then when you look at the community level, so now you're thinking of public health implications, you'd like a vaccine that actually reduces transmission amongst persons, person-to-person transmission. And by reducing transmission, we know that indirectly or directly speaks to actually achieving herd immunity when you've got a sufficient threshold of your population that is vaccinated to actually cut out transmissions or reduce transmissions considerably. And by reducing transmission, you then reduce the burden of disease, leading to healthier communities. People come to work all the time. They put in maximal hours, less sick days, less burden on the healthcare system as well, and less morbidity in the community. However, when we look at COVID vaccines, one of the things to highlight is the vaccines that have actually been authorized or licensed. I think most really have been authorized for emergency use, and the clinical trials that have actually reported on these vaccines have clearly demonstrated that the vaccines are very beneficial in reducing the severity of illness. And we'll look at some data later on. However, with regards to preventing infection, data is still trickling in, and it points to that vaccines actually will, most of the vaccines actually do reduce the rate of infection amongst those that are not infected. And then when you look at transmissions, like for instance, by rolling out vaccines, does it mean that you actually reduce transmissions in the community? Again, that is data that is still being explored. There is data that has been coming in. For instance, if you look at the past 2 weeks or so, there were reports from the AZ vaccine that showed a 67% reduction in infections after 2 doses of the vaccine, as well as an 82% drop in cases of symptomatic disease. And there are some other studies that have been reported as well following the clinical trials, looking at the vaccines that were produced by Pfizer and Moderna. And we know there's ongoing work to look at that. And we also heard from the FDA submission that was on Friday, when they discussed some of the findings from the Johnson & Johnson vaccine trial, that there was a suggestion that the vaccine does, in fact, beyond just reducing the severity of illness, there is a suggestion that it might actually reduce individual infections and transmission as well. But however, this is data that is still being explored, and the confidence is still little. So data is still coming in. But what does that mean? So because scientists are still learning whether or not the current COVID vaccines can effectively prevent transmission of the virus, while it is likely that they do reduce the risk of virus transmission, probably that might not be for everyone, right? So this is one of the reasons why, even if you go out and get vaccinated, it will still be important for persons to continue practicing the standard preventive modalities that have been in place in public. For instance, wearing masks, practicing physical, social distancing, washing your hands, etc. So it is important that even after being vaccinated, you still have to continue practicing these measures. That is wearing your face mask, practicing social and physical distancing, and also hand washing, amongst many other interventions that are there. So let's move on to look at how a COVID vaccine actually works. So before we get into the specific vaccine, let's speak of vaccines in general. So vaccines stimulate the human body's own protective immune responses, so that if a person is infected with a pathogen or with a germ, the immune system can quickly prevent the infection from spreading within the body and causing disease. So in this way, vaccines actually mimic natural infection, but without actually causing the person to be sick. So if you look at the schema or the picture in this, on the slide, you see we've got a picture of the virus there. The body recognizes the virus. It sounds an alarm. It gets in fighter cells, antibodies to come into action and fight the virus. by blocking and destroying the virus, then you cannot be infected. And then what happens with the vaccine? You're trying to mimic this, and you're also counting on some special characteristics of the immune system, like being able to mount that response, be it an antibody response or a T-cell mediated response, etc. And also you're also counting on memory of the immune system, such that when the person is now exposed to the real pathogen, their immune system has a memory, remembers that pathogen, and then the body is ready to actually mount a response to fight that infection. To understand how COVID vaccines actually work and are made, we need to first understand the basic structure of the COVID virus, sorry. So if you look at the diagram or the picture that's on the slide, you can see that you can imagine the virus as being a spherical particle or organism with genetic material inside, and then with spikes on its surface. These spikes are known as spike proteins. So they're described as landing decks for the virus to actually infect a cell. These landing decks have to land on the cell attached to a receptor, and that allows infection to happen, infection to happen. So most vaccines actually focus on the spike protein of the virus. So the immune system is primed or taught to develop antibodies or to develop a response that can actually recognize that spike protein and then set out a chain of events that results in blockage of the virus from further causing havoc in the human body. So for instance, if you look at an antibody response, the antibodies will actually latch onto the spike protein and stop the virus from attaching to the human cell. So a successful vaccine for COVID would teach people's immune systems to make antibodies or to mount an immune response against the virus without actually causing disease. This is a nice schema that actually tries to illustrate what we were speaking on the previous slide. So this shows how a typical antibody response might actually occur. But when you actually think of the immune system and the kind of responses that are stimulated to fight off a virus infection or that are stimulated by a vaccine, it may be more complicated than this. But this is sort of like the antibody neutralization model, and it tries to sort of like illustrate how this could work. But we know the immune system is more complicated than that, and there are other responses that are called into action. So here you have your SARS-CoV-2 virus with those spike proteins. And then when it tries to attach to the human cell, it needs to attach and then uses those receptors, your transmembrane ACE2 receptors, to actually get into the cell. So these receptors are also on the cell membrane. But then with your antibodies, these antibodies are Y-shaped molecules that then come in and latch onto the spike protein, blocking it, preventing it from getting into the cell or preventing it from infecting the cell. Let's spend some time on this slide because now we're going to talk about the types of COVID vaccines that are actually out there. So first, or should I say generally, the different approaches to creating a COVID vaccine, or the different approaches or the different types of COVID vaccines have generally been categorized into four. You have the inactivated type or the whole virus type, and then the viral vector type, the nucleic acid type, and the protein type. But let's start with the inactivated or the whole virus type. So the whole virus approach is whereby you are actually using inactivated or live attenuated subunits of the virus to actually create your vaccine or inside your vaccine. So most vaccines in use today incorporate an inactivated or weakened form of a virus that is not able to cause disease. So when immune cells actually encounter them, they then make antibodies. This is an old and tried, tested method that is actually used in a lot of vaccines that we've been exposed to or that we've got before. So making these vaccines means growing viruses and lots of them, and then you either weaken or kill those viruses in laboratory conditions. And for instance, your influenza vaccines would typically be grown in chicken eggs, and other vaccines are grown in tanks full of floating cells. And these procedures usually would take months to produce like a batch of new vaccines. So one of the limitations really of this approach is that in terms of speed of production, this is a much slower approach. Examples would be like your conventional vaccines for influenza or chicken pox, measles, mumps, rubella. They all fall into this category. Then in terms of the current COVID vaccines on the market, we've learned about the Sinovac and Sinopharm vaccines, and these are vaccines that have been deployed in other parts of the world, for instance, the Middle East and other parts of Africa. Zimbabwe, for instance, started a healthcare worker vaccination program the past 2 weeks using the Sinopharm vaccine. So this is a vaccine, again, we said this approach of vaccine, you're actually using a whole virus, either wholly killed or attenuated or weakened, and then you're using it to actually create a vaccine so that you stimulate the immune system. Let's move on to the next type. So the next type that we see there is your viral vector vaccine or the viral vector approach. So when you think viral vector, one needs to think a vector as a mode of transportation. So you're using the virus as a mode of transportation. So viruses are very good at getting into cells. I think we all appreciate that. And since the 1990s, researchers and scientists really have been investigating how to use them to actually deliver genes into cells so that they can use those genes to immunize people against diseases. So with this approach, what you're actually doing, you're adding the spike protein gene to a virus. And one of the common viruses that is used in this approach is something known as an adenovirus. But basically, it would be a virus that is whose genetic makeup has sort of been tempered with so that it does not replicate in a human being, or it does not cause disease, or it does not multiply in a human being. Then this virus slips into cells and unloads the gene. And because this adenovirus has been tampered with and it's missing one of its replication genes, it cannot multiply, and therefore it is actually safe. So with this approach, that's where you see your AstraZeneca vaccines. I know we know a lot about these vaccines because they were trialed here in South Africa, the Oxford AstraZeneca vaccines, and also the Johnson & Johnson vaccine, And then moving on to the next approach, we have the nucleic acid approach. So this is where you have genetically engineered plasmid containing either DNA sequences or messenger RNA that contains sequences for the specific antigen, in this case, the SARS-CoV-2 spike protein, are actually delivered to cells, And then this protein can actually stimulate, or should I say, you're not delivering a protein, but you're delivering genetic instructions for building a viral protein. And then when those genetic instructions get in the cell, you're actually using the cell's own machinery to actually build up those proteins. And those proteins then stimulate the immune system to mount a response. The downside of this approach is that these vaccines have to be kept and stored or transported in ultra cold temperatures. For instance, you're looking at super cold freezers like -70 or -20, and these are usually freezers that you don't find in routine care, but perhaps in research settings or settings that are like that. And this can prove challenging for countries that don't have specialized cold storage treatment, particularly here in sub-Saharan Africa. And then the last approach that we're going to look at before we move on with the slides is the protein subunit approach. So these are vaccines that use a protein fragment or a piece of the protein of the virus. So some vaccines some of those vaccines that use those pieces of proteins stimulate the immune system because for the immune system to mount a response, it doesn't need to see the whole virus, but it needs to see those special epitopes or those special immunogens that actually then stimulate it. These pieces of protein can't cause disease because they're not the actual viruses, but they can still show the immune system what a virus would look like. So if the immune system sees that, it thinks that it's seen the virus, then it mounts a response. So armed with this understanding of the different types of COVID vaccines, let's move on to then look at how vaccine research is generally conducted. This also speaks to drug development as well. So these always begin as thousands of ideas in the laboratory, in academic institution, in biotech or pharmaceutical companies. And usually this is a graduated approach so that only the best ideas make it to the next step. And by the time you get to your human trials, you would have gone through research in the lab and also research in animal models. As you can see, you've got different grades of animal models as well, including non-human primates just before your human trials. And then when you look at your human trials, this is phased as well research that is graduated. For instance, you begin with very small trials in very healthy people, whereby you're evaluating safety, determining dosage, identifying side effects, and approximately 70% of drugs, this is from the FDA, will then probably make the stage and move to the next one, which is a phase 2. So with the phase 2, you now have got larger numbers, but now you want to actually test or check or evaluate if your product actually does what it's supposed to do. So for a vaccine, you're now testing for things like immunogenicity, maybe effectiveness is an overstated term there, but you're looking at things like immunogenicity, what kind of responses does it stimulate? Are those responses sufficient enough to prevent against infection? And then you're further evaluating on safety. And then after this stage, the ones that actually pass this stage then move on to phase 3. And as you can see from that diagram, it says about 33%, 1 in 3 would actually move on to the next phase. That 1 in 3 that moves on to the next phase, that's your phase 3. So with your phase 3, you now sort of like have an idea that your drug or your vaccine will actually perhaps do what it's supposed to do, like it stimulates those robust immune responses, it blocks the virus, it is relatively safe. And then you move on to much larger studies known as phase 3 studies, whereby you want to actually confirm effectiveness, continue to monitor side effects, but at this point, you're now doing a trial in much wider populations. And you've seen from the COVID vaccine trials that sample sizes ranged from as little as 5,000 to as much as 45,000, for instance, with the Johnson & Johnson trial. So it's much wider populations with comorbidities, and now you're looking at different types of populations across the globe. And then in some designs, you're actually comparing against treatment and collecting information. And then those drugs that actually pass, or those vaccines that actually pass the stage, they're the ones that eventually become licensed. And then for licensing, they need to also meet some minimum criteria as well. For instance, the effectiveness needs to be above a certain threshold. The safety really needs to be good. And then the durability as well, because it's of no use developing a vaccine or a drug. Maybe no use is an overstatement, but it will be more useful to actually have a vaccine that is very durable, that lasts much longer, that will have impact for a much longer time than a vaccine, for instance, let's say that would only be effective for, let's say, 2 weeks or a month. So durability is quite important. And beyond that, this testing never stops. So you've got phase 4. This is where you actually do your post-marketing surveillance. The vaccine is now authorized or licensed, but you still have to collect data. You still have to collect data for safety, and then you're now looking at other risks and looking at other benefits. You're also looking at effectiveness as well on a wider level. And we know that in clinical trials, you're usually limited with the numbers of persons that you actually enroll in that study. But then when you move into the real world, what might have been very rare in a clinical trial, you might actually start to see that in a wider population. Imagine a rare event that occurs in 1 in a million. If you roll out that drug in the population, let's say you're rolling out that drug in a population of vaccine, in a population of 55 million, the occurrence then jumps up to 5 in 50 million. Yeah, like 5 in, I think 50 in 50 million. Yeah, 50 occurrences in 50 million for 1 in a million. So you really need to continue monitoring safety. So the monitoring of safety doesn't stop. The monitoring of safety doesn't stop. We've got a slide towards the end that actually speaks to this whole parallel process that was followed when developing the COVID vaccines. So we've spoken about this. Now let's look at vaccine effectiveness. So I thought rather than going through the different trials and studies that looked at effectiveness, this is a very, very nice summary of the different vaccines that are there and their ranging effectiveness. So for instance, if you look at the extreme left, this is basically illustrating effectiveness from your flu vaccines. So again, this is coming from CDC affiliated organization, the AFP, and this slide is created for the public. 2014 to 2015, you're looking at 19% effectiveness. 2010, 2011, 60%. 2019, 2020, looking at a 45% effectiveness. But if you look at the COVID-19 vaccines, really the effectiveness that has been demonstrated in clinical trials has been, I wouldn't say astonishingly high, but it has been impressively high. It has been impressively high. And then also, if you compare with other very effective vaccines, like your vaccines for rubella, measles, polio, you also see effectiveness that is above 90%. So we are in a state where we have effective, safe vaccines. We'll look at some of the safety data. And really what would inform a community or a country in terms of which vaccine to use depends on a different number of things, which my colleague Azwi will speak to later on. Moving on, there are some studies that I would like to highlight. So, okay, the slide eventually moved. We've all heard about the Johnson & Johnson vaccine. We looked at the different types of approaches for building up or for creating vaccines. So the Johnson & Johnson vaccine uses a viral vector approach. It uses a viral vector approach. So this vaccine was shown to be very effective against preventing severe disease. The effectiveness was around 85%. And then it also demonstrated complete protection, which is 100% protection against COVID-related hospitalization and death as of day 28. In the study, it was a multi-center study, global study, whereby close to 44,000 participants were actually enrolled. Close to 500 cases of COVID were accrued in the study. The study was conducted in the United States, Argentina, Brazil, Chile, Colombia, Mexico, Peru, and South Africa. South Africa actually contributed 15% of the study population. So around 6,500 participants were from South Africa. And during that time of the study conduct, the prevailing or the predominantly circulating strain was the strain from the B1351 variant, which I saw later on in the afternoon. Later on in the afternoon, there will be a talk with regards to that. So this vaccine showed good effectiveness against that strain, and it showed very impressive effectiveness when you looked at severe disease or when you looked at death and hospitalization. Also, the vaccine had an acceptable safety profile in line with the adenovirus 26 platform. As we said before, this is a viral vector, and with the viral vectors, you actually use a virus that is non-replicating, or it's incapable of replicating. And this is not the first time this kind of platform was used for a virus. For instance, this platform, ART26 platform or adenovirus vector platform, has been used for a number of vaccines. For instance, vaccines that we've used in HIV, these are still in development. Then vaccines that have been licensed for Ebola, vaccines for some of the hemorrhagic fevers, some of the filoviruses, and for RSV as well. So this platform has actually been rolled out already in more than 200,000 people and has been shown to be safe, the adenovirus platform. And then looking at this trial in particular, there were no safety concerns. The vaccine had an acceptable safety profile. Overall, the serious adverse events that were reported were similar between the placebo and the vaccine groups, and they were really low. They reported an SAE rate, a related SAE rate of less than 1% across both groups. And there were no concerning safety signals as well. And the advantage is that just like the AstraZeneca vaccine, these viral vectors vaccines, they're stable at normal refrigerator temperatures like 2 to 8 degrees, and also in room temperature for up to a certain period. For instance, up to 2 hours in a vial or syringe, and this can go up to 6 hours if the vial is not punctured and if the temperature is actually controlled. And this is actually realistic logistics for sub-Saharan Africa. Another example of a viral vector vaccine that is important to know about is the gamma COVID vaccine, also known as Sputnik. It also uses the same approach, but it's sort of like a modified approach whereby they use what is known as a heterologous prime boost strategy. So what they're actually doing here is, if you think about it, an effective vaccine usually requires more than one-time immunization in the form of a prime boost. You go, you get your first vaccine, which primes, and then you go and get another one, which then boosts. So traditionally, these same vaccines are usually given multiple times as homologous boosts, right? Just like you think of some of the childhood vaccinations. But however, when it's a heterologous prime boost, it means you actually doing it, giving different types of vaccines containing the same antigen. So this is quite an impressive approach. And from other vaccines that have been used before that use this approach, they've been shown to actually have very good, robust responses, very good, robust responses. And it's actually an effective approach for generating a powerful and a much longer lasting response. So this is a vaccine that was developed by this institute, and it's known as Sputnik as well. It uses the viral vector. It was reported recently in the Lancet, and they also showed very impressive effectiveness rates. And when you look at these nice Kaplan-Meier curves, you actually see that they start to sort of like separate between 10 and 20 days, probably day 14 or so. And that's what's been seen with a lot of vaccines. That's when the effectiveness starts to sort of like mount in and show. So what it means, or what this speaks to, is even if you've been vaccinated, it doesn't necessarily mean that you are protected completely from COVID or from COVID disease. Remember, the protection is not 100%. Then two, also, this protection, you start to see it a bit later after vaccination. Really, 14 days is what a lot have reported. Some have reported 21 days, some 8 days. Then for your severe disease or so, I think protection has been seen as early as day 7 with some of the trials. This is just a picture from 2 weeks ago, the Sinopharm vaccine that we spoke about being delivered to a country just north of us. And this is just a schema that I'm sure you've all seen, because it's all over the place, really, comparing the different vaccines in terms of efficacy, the trials, the doses, and storage. And what I would like to highlight is that most of these are actually 2-dose vaccines. And then when you look at the J&J, this is a 1-dose vaccine, but I'll just want to emphasize that there is actually a 2-dose J&J vaccine trial that is ongoing, that is currently ongoing, including in South Africa as well. But the current one that has been reported in clinical trials and the one that is used in Sisonke, it's a 1-dose vaccine. So before we close, let's touch on safety. What are you expected to, or what are the expectations really after getting a COVID vaccine? So quite a lot, if you've read through the scientific literature, or if you've spoken to someone who had a vaccination. I spoke to a friend who had a vaccination recently. They had to think twice about continuing doing their call on the day of vaccination. And they sort of like called to say, hey, I don't understand what's happening. I feel sick. So it is totally normal to experience side effects during the first few days, about 1 to 2 days after vaccination. This is something known as reactogenicity. It's a sign of your body building up immunity. And here is what you may actually experience. So on the arm where you got the injection, you might feel pain, there might be swelling, there might be erythema or redness, induration or hardness. But these are usually self-limiting symptoms. They go away on their own quickly within a few days. And at times, if the pain is a bit too much, you may need to take painkillers like Panadol or Prof. and also sometimes even apply a wet cloth over the injected area. And it's quite not common. It's actually rare for this to actually worsen such that you need medical attention or admission to a hospital. It's quite rare. And then systemic signs and symptoms that you might experience. You might experience fever. This is seen quite a lot. And I know growing up and watching my mom taking my young brother to go get a vaccine, I'll know that I would have a restless night because the young boy would be so hot and restless, etc. So I sort of like always think about this when I see reactogenicity. Then things like chills, headache, tiredness, joint aches, muscle aches, nausea. And then coming to allergic reactions. Allergic reactions are actually not common. They are actually not common, and we'll look at some of the data. And then your severe allergic reactions or even life-threatening allergic reactions like anaphylaxis are actually very rare. But we'll look at some of the data in I think this is like the third last slide or so. So this is data from vSafe. So it's a system that was set up by the CDC and the FDA for passive reporting of side effects for persons that have been vaccinated from the COVID vaccines. So this is data from the 2 mRNA vaccines that they had rolled out, the Pfizer BioNTech vaccine and the Moderna vaccine. This is data from mid-January. By then they had vaccinated, by then they had reports from about 22 million administrations. And when I checked this database last week, they'd gone up to about 55 million. So this is quite interesting data. Of course, there are limitations with self-reported data, but it actually starts to give insights. So what we know about mRNA vaccines is that they're quite reactogenic when you compare them to like your viral vector vaccines. And as you can see from this slide, 70% actually had pain, 1 in 3 had fatigue, about 1 in 3 as well a headache, 1 in 5 myalgia, about 1 in 10 chills, fever, swelling, joint pains, and nausea. And this sort of like went up. So this is overall, and this sort of like went up when they got the second dose. So reactogenicity is common, and with the mRNA vaccines, really, it's been shown to be a bit more prevalent, or it's a bit more incident than your other approaches. So it is common, and if you are going to get vaccinated, do expect some reactogenicity. And the good thing about reactogenicity, maybe good thing is not a good word, but the reassuring observation about reactogenicity is that it is usually self-limiting and most of the time will not require treatment. But when it requires treatment, it's usually over-the-counter medicine that will go away. And I see on the chart, someone is actually alluding to this, and a relative actually had a fever and they could not go to work the following day after receiving the J&J vaccine. Yes, this is not abnormal. This is a sign of your immune system in play. Yeah. And then last but not least, let's look at the most severe of the allergic reactions, the so-called potential life-threatening allergic reactions. So anaphylaxis is potentially life-threatening. This is a very severe allergic reaction, and they've also looked at this data in the vSafe database. So from the Johnson & Johnson trial that was actually conducted in South Africa and globally, over 44,000 participants, no anaphylaxis was observed. No anaphylaxis was observed. This has been reported from those 2 mRNA vaccines that have been rolled out in the US to occur at a rate of between 2.8 to 5 per million doses of administration. So again, this is really rare. And what has been seen is that participants or persons that actually experience these severe allergic reactions Not all, but quite a number would have had a history of allergy to a vaccine component, allergy to food, or allergy to some injections, etc. So hence you'll see, even when the rollout begins, or even in the ongoing Sisonke study, before you get vaccinated, there is always a check to say, do you have an allergy history? Have you reacted to vaccines before? Or have you reacted to contents of that are in the vaccine before? And if you are that such of a person, depending on a risk, you can be vaccinated under very controlled conditions, whereby an emergency trolley is just there and you've got your healthcare providers ready to assist or treat if there are issues, and a much longer observation period. Usually after vaccination, you're observed for 15 minutes, but if you've got a history of allergy or a history of some reactions, the observation period is even made much longer. It's even much longer. And then if you've really got that severe allergy history, sometimes you can be turned away and asked to see your primary care provider or even see an allergy specialist. But this is not common. This is not common. As you can see, this is quite rare. In acknowledgement, I snipped off some of the slides from the core VPN. It's a village that we work with, and also snipped some from the National Department of Health website, NICD, some of the resource centers that are shown there. And thank you very much for your attention. I will now hand over to the moderator. I think Dr. Azwi Takalani will take over from here. Thank you very much.
Thank you very much, Dr. Simba Tavuka. My sincere apologies. I printed an older version of the program with a bit of an error. Thank you very much for your contributions. That was Dr. Tavuka dealing with the COVID-19 vaccine science, development, and safety. And I now immediately hand over to Dr. Azwi Takalani. And Dr. Azwi will deal with the COVID-19 vaccine workplace program considerations as we have it on the program. And she has an MBCHP Med, FCPHM, and she's a public health medicine specialist, a safety physician, medical monitor, also with the Sisonke open label trial. I hand over to you immediately just to check if your audio is okay.
Hi, and thank you, Ashraf. Can you hear me?
Great, we can, and your slides are visible. Please proceed.
Okay, you can, I've actually got two monitors. Can you see my slide, my presentation mode slides, or the other version?
The presentation mode, I think it's perfect for our audience.
Okay, perfect. Okay, thank you very much, Ashraf and the NIOH team. It's really such a privilege to be able to present to the occupational health practitioners on this platform. You've introduced me quite well. I will not go through that again. And I just wanted to say, Dr. Tavuka and I thought it would be important for the audience to really understand the vaccine science and immunology and the vaccine candidates out there quite well before we go into some considerations of workplace programs. And that's why you see that he's taken a bit of my time, but that was intentional. I'm just gonna move on. Okay, sorry. Right, I'll move to slides. Same conflict declaration as Dr. Tavuka. I'm a safety physician and medical monitor on ongoing COVID-19 and HIV vaccine trials. And some or most of these trials are actually sponsored by industry. And I also work on the Sisonke trial, which is using the Johnson & Johnson investigational products. So I have really divided this talk into two phases, some consideration for the preparation phase and the operational phase. Of note, these consideration of a vaccine, of a COVID-19 vaccine rollout program in the workforce. And of note, these will really just be high-level considerations. And we would really like to encourage the occupational health practitioners on this platform to complete the vaccinators course that's on the National Department of Health Knowledge Hub website, because there are more detailed and clinic considerations. And also we will circulate to the NIOH team a tool, a vaccine clinic algorithm tool that deals with the details of a vaccination that you need to consider when you're setting up a vaccination program. I have to acknowledge that it's based on the J&J vaccine products, but most of the information there actually can be, the vast majority can be applied to multiple of these vaccine candidates. So starting off with the preparation phase, I just wanted to first put in context where work-based programs are in the national rollout of vaccines. So there's phase 1, which is vaccination of frontline healthcare workers, in a way, because these vaccination centers are also based in hospitals, or most of them will be based when the rollout actually starts. It is a workplace-based program in a way, but mostly the considerations that we'll be talking about are what most of you will have to deal with when phase 2 and phase 3 of the rollout comes into play, where all essential workers and persons in congregate settings may need to be vaccinated, as well as vulnerable persons who are people over 60 and people over 18 with comorbidities. And then finally in phase 3, which is mostly persons who are over the age of 18. So in these 2 phases, there will be vaccinations happening in public facilities, in vaccination centers, such as in GP rooms or pharmacies. And there will also be outreach programs that will aim to reach people in congregate settings. And the important one for us, of course, is work-based vaccination program. So for now, there isn't total clarity as to what this will mean. Will we partner with the National Department of Health? Will private purchase be easily attainable? But I think the considerations, regardless of the background logistics, are really still the same. So when thinking about covering a majority of your employees very quickly, a lot of occupational health practitioners are really wondering if they shouldn't just consider putting mandatory vaccination policies in place. But if you really look at the current legislative framework, it is quite unlikely that blanket mandatory vaccination policies will be easy to put in place in the South African context, unless, of course, the Ministry of Health makes additional proclamations to the existing legislature. So starting off looking at the Bill of Rights, and enshrined in there in Section 12 is the freedom and security of the person, which actually goes to explain that everyone has the right to bodily and psychological integrity. And this includes the right to security in and control of their body. And then if we look at the National Health Act number 61 of 2003, Section 7 deals with informed consent and really states clearly that a health service may not be provided to a user without that user's consent. And then there is a reference also in Section 7 that says, unless of course that treatment may avert a serious risk to public health. And however, in just viewing the spectrum of legislature, it may be quite onerous on an employer to prove that this is the case, to hang the reasoning for forcing an employee to vaccinate on that one, unless, as I mentioned earlier, the Ministry of Health makes further proclamation in this regard. And then finally, if we look at the Occupational Health and Safety Act, it provides or it requires the employer to provide health and safety at the workplace in reasonable, practicable measures. So looking at the spectrum of the legislature and trying to see where mandatory vaccination policies could fit, it really looks like the net balance is these will probably not be the case. These will probably not be possible in the South African context. So then we need to look at other options as occupational health practitioners. In the absence of making it mandatory, what should we be doing? And we should really be capacitating our employees to make informed decisions about vaccination. or the vaccines, to vaccinate or not to vaccinate. And really this process should start right now or soon if it has not already been initiated at your workplace. And the capacitation of employees is really going to take multiple stakeholders coming together to form a policy background and also to create an environment that allows uptake of these vaccinations, starting off with management buy-in. So management will really need to buy into this program and support or partner with the National Department of Health, or if there's private purchasing that's going to be allowed, then in line with those, the governance of procurement of COVID-19 vaccines, you need to have these vaccines in your workplace as supported by management. And then of course, the employees need to really understand what the vaccine is about, the side effect profile, the efficacy, and the safety of the product. And I'm going to speak a little bit about this in the next slide. The occupational health practitioner is then obviously central in this regard because they are the ones who are going to be putting all these stakeholders together to ensure that there is buy-in from management and to ensure that the employees actually understand what's happening, and also to rope in human resources. As Dr. Takuva has explained, we think that there will be reactogenicity after vaccination. And so there shouldn't be punitive measures and people being forced to take the annual leave because they feel a bit feverish the next day and are unable to come to work. And of course, we need labor representatives to also fully understand what the vaccine is about. So on my next slide, I think it's just a summary of everything that Dr. Takuva has gone through, and basically saying that occupational health practitioners really need to bring vaccine hesitancy immunity into the workplace. And this can only really be achieved if the employer and the employees understand the vaccine science. That's how the vaccine works and how they developed, if they understand the efficacy as well. And this has to be tailored to the vaccine candidates that will be rolled out in your workplace. And they need to also understand the continuance of non-pharmaceutical interventions, as Dr. Takuva has already mentioned. So just because you're vaccinated does not mean you no longer need to wear your mask or practice physical distancing and hand hygiene and so on. They also need to understand the safety profile of each and every single vaccine candidate that will be available in your workplace, and most importantly, the common reactogenicity profile. And also the occupational health practitioner will have to really contextualize all serious adverse events. So for example, Dr. Takuva has explained that anaphylaxis happens between 2.8 to 5 in a million. So people should understand how rare and exceptional it is for this, for these sort of severe adverse events to occur. And obviously to understand the regulatory processes that go in place before a vaccine is actually licensed and approved for a rollout. So all these four key things will actually be what will determine if there will be uptake in the workplace of the vaccine. However, even when we are trying to optimize the uptake, we need to really think about how we're going to manage exemptions. And exemptions can range from being totally legitimate to being based on unfounded myths. So the previous slide dealt with how to probably debunk some of those myths by explaining the science and the clinical presentations of the vaccine. But then in terms of exemptions, there will probably be medical exemptions. For example, a person who may have a severe allergic reaction to the vaccine constituents, so that person probably cannot be vaccinated in a work-based place setting. That person needs to be referred to a specialist who needs to do further assessment and will probably be only vaccinated under controlled settings. That is where all the resuscitation equipment is ready and there's sufficient staff to ensure that they can support this sort of person should they have a reaction. There may be religious exemptions as well. And I think the important takeaway message here is to explore the grounds if the employee is willing for opting out on religious grounds, because some of these may actually be myths and need to be debunked. And then, of course, like I said, mandatory vaccination policies may not be easy to implement in the South African context with the current legislature. And so some people may opt out of actually being vaccinated. And so how are you going to manage all these exemptions? And again, it's education, awareness, promotion, and health promotion. But when a person opts out, you need to document clearly that the vaccine was offered. You need to document the employee's decision. And if it's in a contingency plan, so say, for example, it's someone who has a severe allergic reaction, you need to document that you referred them to their specialist for further assessment and vaccination in a controlled setting. It is quite likely that as you progress in the workplace program, people who may have initially refused to take the vaccine or have been hesitant, as they see more people taking it up, they may become more keen to actually have the vaccine, and you may have quite an onerous task proving that you offered them the vaccine if you haven't documented appropriately. That's not to say if someone changes their mind, then they should not be vaccinated, but you could find yourself in a litigation situation where it seems like preferential job categories were probably offered the vaccine. And then looking at some considerations for the operational phase quickly. So the first consideration that I wanted to look at was vaccine scheduling. And I think Dr. Takuva took us very nicely over the reactogenicity that actually some people will experience. And this varies from about 25% to 70%, depending on the vaccine candidate. So obviously, once you roll out your program in the workplace, you want to optimize coverage as soon as possible, but you also don't want to bring operations to a halt with vaccinating everybody on the same day, and maybe 30% of your staff then needs to be off for the next day or two. So in considering vaccine scheduling, consider a staggered approach. So not vaccinations every day, possibly skip some days, or schedule staff in a way that not everybody in the same job category and shift are being vaccinated at the same day. Of course, that is taking into consideration your operational needs, and you know best, your company best. And then, of course, as I mentioned, and Dr. Takuva explained so well for us, the reactogenicity profile. People may need to take days off post-vaccination, and also, especially if you're looking at the 2-dose regimens, because there is usually more enhanced reactogenicity after the second dose. And then some considerations for vaccine storage and distribution. And this table should look familiar. It was also in Dr. Takuva's slide. And here, I just wanted to highlight the column that says cold chain. And you can see that the cold chain requirements vary from ultra cold right at the bottom for Pfizer, which is -70, -20, and all the way to 2 to 8 degrees. So you have to know your vaccine candidate, you have to know the cold chain requirements, and it may even be tricky because depending on vaccine availability, you may be even using different vaccines at different stages of your workplace program. So you need there to ensure that you have reliable refrigerators and freezers, appropriate temperature monitoring devices, a good protocol on how you're going to manage vaccinations that have been exposed to temperature excursions, a good vaccine inventory management system, because as we proceed, it may become quite the lucrative lucrative business to sell COVID-19 vaccines. So you want to make sure that you have a good inventory management system where people aren't able to access the vaccines, or you are at least able to account for all vaccines at the same time. And you may need to think about some transportation logistics. So you may be working in a company where there are different plants, and all people, you may want to go to different plants and transport the vaccine with you. What about cold chain requirements during the transportation, and also keeping the vials safe so that they do not break as you transport them? And then finally, and most important to me as a safety physician, is the adverse event following immunization reporting. And this is really very key, because as we gather this information, we are able to feed it back to your workplace, feed it back to the public in terms of just which adverse events are really common, and just how many people actually don't have severe events or have, or complications due to immunization by the COVID vaccines. I think most importantly, COVID vaccine AEFIs are going to be reported via the National Department of Health EPI systems that already exist. So all reporting needs to be aligned with this reporting. And even when we started with the Sisonke trial, I think we noted that a lot of occupational health practitioners were actually reporting via their usual channels and were not aware of the National Department of Health channel of reporting AEFIs. And so this is going to be dual reporting so that it can feed into both systems, because there are also regulatory requirements for this reporting. So the National Department of Health, currently their system is paper-based, but they are developing an electronic tool which will make it easier for reporting. And currently, if there's anybody who is working on the Sisonke trial and vaccinating healthcare workers, we do have an electronic ECRF that each and every single vaccinee receives prior to vaccination, so after registration. And we encourage reporting through that ECRF, or to call our safety desk, which is a toll-free number, or actually for people to report via the usual National Department of Health system, which is their healthcare worker completing this form and sending it to the appropriate places. And with that, that was a whirlwind through some of the considerations for a work-based program. And I'm going to end there. Thank you, Ashraf.
Thank you very much. If you could just share your presentation. So that is Dr. Azwi Takalani, and she partnered with Dr. Simba Tavuka. The two of them work as medical monitors and safety physicians with the COVID-19 Prevention Network and the HIV Vaccine Trials Network. Apologies. The HVTN conducts all phases of the clinical trials, from evaluating experimental vaccines for safety and immunogenicity to testing vaccine efficacy. Leveraging on the infectious disease expertise of the existing research networks and global partners, the NIAID formed with the COV-PN, whose mission is to conduct phase 3 efficacy trials for the COVID-19 vaccines and monoclonal antibodies. Simba and Azwi will discuss the COVID-19 science, development, safety, as well as occupational program considerations. With that, I move over immediately to Dr. Sipho Senabe, who is the Chief Director for Human Resource Development and Employee Health and Wellness in the Gauteng Department of Health within the Gauteng Provincial Administration. Dr. Senabe, just to check on your audio and video connection, if you could just check that with me. In the meantime, a reminder to everybody, we will have a red cap survey after the session is done, the webinar is done, for all of you to give feedback, as well as tell us what other new topics you may require. for us to run on our COVID-19 webinar series here from the NIH. I see Dr. Senabe is live. I can see his video. If you could just check your audio.
Yes, good afternoon, everybody. I don't know if this is, I'm audible.
Very, very good. Thank you. Please proceed. Will you be sharing your presentation?
I have sent my presentation to the secretariat there. If they could upload for me, I would appreciate.
Okay, I will arrange with my colleagues in the IT section, and I'm going to ask Tabani and Glenn to just assist in uploading Dr. Senabe's presentation.
Alternatively, I could try it from my side if
it works, check, yeah. In the meantime, just as an administrative reminder, if you could maximize the slides, Dr. That's perfect. So as a reminder to everybody, please type your questions in the Q&A box and our presenters and panelists will answer them there. I hand over to you immediately, Dr. Senabe.
No, thank you very much, Chairperson, and thank you to the fellow panelists and the attendants to this webinar. The perspective I will be sharing really will be on the COVID vaccine for workers in general. I will be sharing an occupational health perspective, largely from a legal and ethical perspective, but a bit about the practicalities of how do we ensure that we have a vaccine trial in an occupational setting and how it's going to be affecting the rest of the vaccine program. The first thing I just wanted to say is that in Gauteng, whilst we are working directly mainly with health workers and workers in the public sector, we have been collecting data on health workers even in other sectors, and particularly that we have about 11,833 health workers who are employed by the Department of Health who are infected with COVID over the first two surges. In the past 24 hours, as of yesterday, there was only 2 who demised, so it does still impact on mortality. And at a larger level, we have 98 workers to date who have actually been, who have demised. We have had a few who are in the private sector who are also not as infected as the public sector, but it is about 3,449. Then you would have the others in the public sector and in national government departments and in municipal workers. What is common with all of the with all the categories is that those workers that are in the public sector are seemingly the most infected, and to a large extent because these are the essential service workers, police, municipal workers, health workers in general, both in private and public, those who are actually facing the public, and of course, less so in the other sectors. If we want to drill at a more detailed level, Gauteng has about 1,686 of those who have been hospitalized, and of those 1,518 were discharged. As indicated before, 98 died, and there were those who were in hospital and transferred to other facilities. Of note is that most of them are nurses for obvious reasons, that we are in a predominantly women or female dominated industry by virtue of the helping sciences, and with those who are medical practitioners and support function employees infected the least. This is just to emphasize that we've gone through 2 waves or 2 surges in Gauteng and in the rest of the provinces. And the context of this discussion is to assist us to introduce the intervention of COVID vaccination to avoid a third surge. This slide still speaks to those who were ever infected, and that Gauteng had the largest number of employees who were ever in hospital or hospitalized as compared to all the provinces. And this has implications for COVID surveillance, post-COVID surveillance. And now in the context where there is going to be vaccination of some of these people who were ever hospitalized, it is going to be a combination of post-COVID surveillance and post-COVID, or what has been earlier indicated as post-marketing surveillance after vaccine administration has been effected. I'm going to skip this slide because it has, been very eloquently presented by the first two speakers. Suffice it to say that we are on a phase 3, specifically phase 3B of a clinical trial that is following the J&J ensemble clinical trial, which was of stage 3A, led by the Medical Research Council, approved by SAPRA as a regulator, and it was clear that J&J's a vaccine are safe and efficacious. Now in stage 3B, which was really affected or really demanded on all of us because there was an earlier planned AstraZeneca vaccine, which was going to have the first dose and second dose administered to workers. And because of the deliberations about the strains, the specific South African variant, we had to actually change this, as all people know, so that we can be able to use the J&J that is particularly efficacious against this South African variant. And as a result of that, we needed to make sure that this vaccine is registered as a prerequisite of registration would be that a phase 3B study should be actually conducted. And as the two speakers had said before, this is what we are doing now. At the same time, we are using this for health workers to access this vaccination earlier on. And this phases have been shared before, but specifically with regard to the workplace, is that phase 1, we are targeting 63,000, which is about 21% of 300,000 doses, to target a original population of 1,250,000 health workers, and also 105,000, which is about 21% of those 500 doses that were allocated for health workers in the private sector, in the public sector. And as we have seen, we are getting these vials or dosing in drips and drops, rationed in both the public and private sector. The Gauteng population of this 1.2 million is 215,000 employees, both in the public and the private sector. It is about 82,000 in the public sector and another 100,000 plus in the private sector. At the end of phase 1, we are expecting that we will have had a registrable J&J vaccine with SAPRA, so that we can be able to go to phase 2. And phase 2 again will speak to the priority population of essential workers. It is now being revised that it is not essential workers just as defined by the Department of Employment and Labor, but rather it should be a risk-based categorization and prioritization based on those workers who are most at risk. And those are the workers that are, whose function would be, population phasing, as indicated in my first slide. And those will be mainly public servants in municipalities, those who are in the uniform services, health, those who are also in the transport sector, the retail, according to the category of risk. There is a group of colleagues in the occupational health setting and in the those in organized labor, to ensure that there is consensus about this risk categorization. Then there will be people who will be those who are vulnerable populations, and again here there will be an overlap between these people and those who are workers. And the most consistent consensus on this is age, in terms of those who are 60 and above, and those with comorbidities. We are refining and fine-tuning this to also include other socio-demographic profiles of people, like people who are in dense populations and those who are in overcrowded populations. And so that we can be able to also prioritize those that cannot physically social distance, those that do not have a necessary water and sanitation, and those who are not able to use optimally the option of non-pharmaceutical interventions. And of course, phase 3 is where we are targeting the rest of the population. So we hope that in the phase 2, that the workers will also benefit according to the risk where they come from. Now, this is where I just want to lift the whole issue of the ethics, the law of vaccination, of as we have discussed so far through clinical trials, of vaccination as regulated by the Occupational Health and Safety Act and associated regulations, particularly the hazardous biological agents regulations, and also vaccination as a public health good. And this is where the discussions is going to be a bit slightly different from what some of my colleagues have said before. But the important thing is that this phase 3B clinical trial, which has been approved by the KwaZulu-Natal Ethics Committee, on a J&J vaccine that has been proven to be efficacious, that is led by Sisonke, and that is introduced in the health, in their workplace, has implications in terms of how we categorize COVID-19. Before vaccination, COVID-19 or SARS-CoV-2 virus was classified as a category 4, as classified in the hazardous biological agents regulations. In other words, it's an agent that is hazardous to both workers and the public, that is infectious, that spreads to all people, for which there is no known treatment or even prophylaxis. Now, the introduction through a 3B clinical trial, after a 3A clinical trial that shows effectiveness, changes that classification from class or group 4 to group 3, which is a specific biological hazard for which we now know has an effective prophylaxis. And so it then shifts how we are going to be looking at this, because it then provokes the compulsory nature of worker participation as provided by the occupational health and safety and the vaccination programs. and the hazardous biological agents regulations. In other words, legally, it is compulsory and it is obligatory for an employee to comply with lawful and reasonable instructions of an employer. And that is provided by the Occupational Health and Safety Act. However, there is also the independent duty of the employee to ensure that they also have their own and other and health and safety of other health workers, and including the patients or the people that they actually serve. So these two are the two issues of the dual impositions by the Occupational Health and Safety Act on the employee. Of course, on the employer, as indicated by our colleague before, Azwi, that yes, the employer has to, within reasonable, passing the reasonable employer test, make sure that there is a healthy and safe working environment. But the Act also has these two obligations on the employees. If I were to try and dramatize it, should we allow an ICU nurse to get into an ICU who had actually refused to be vaccinated on religious grounds, as an example, knowingly that it is going to be having a potential impact on infections on other employees and on all patients? It is an ethical question over and above what the Occupational Health and Safety Act provides for them. However, there is an important understanding also of the vaccination as a public health good. That phase one of the vaccination program, which is now fused with the early access study from Sisonke, it is actually there to safeguard the health system by making sure that our health workers are actually protected against further transmission of SARS-CoV-2 virus that causes COVID. And I think this is the crux of what I just thought I would need to lift. Furthermore, I think colleagues have indicated before about the constitutional right to bodily and psychological integrity, and also to secure and control their own bodies. All of us, including workers, have this right. However, insofar as the right to not being subjected to medical or scientific experiments, so the Constitution is very explicit that we cannot force anyone to be in the Sisonke clinical trial, as an example, to apply the Constitution directly. However, the Occupational Health and Safety Act obligates the employer and the employee to be involved in a medical surveillance program where vaccination is offered reasonably and practicably by the employer without risk to other employees and to patients and the public that we serve. And I think this is the type of sort of tight legal interpretations of what the Constitution says on one hand and what the Occupational Health and Safety Act is saying, and they are not contradictory. The Occupational Health and Safety provides for a medical surveillance program. The Constitution protects against being forced to be involved in a clinical trial or a scientific experiment. And of course, in the context of what we are doing now, the adverse events that will occur as a result of the early access study in a work environment, in the health sector in particular, presents a particular a challenge in terms of adverse events that must be managed as both the outcomes of a clinical trial. They must be managed as part of the Occupational Injuries and Disease Act, which basically would say that if you have an adverse event, it will also be an occupational injury or disease. And leave management must be managed as a COID-specific leave. In other words, leave because you are compensated for that particular, for those particular days. You are not going to be obligated to use your normal leave, and that you will also be able to not use your own resources for clinical management of those adverse events. And then the third consideration insofar as this is concerned is, should the Sisonke clinical trial end somewhere in March, and now we are continuing with the vaccination, it would be then taken as a vaccine program for which we will then be managing those side effects as post-marketing surveillance, as has been indicated by our colleagues before. So my concluding remarks are as follows: that the vaccination of employees in a phase 3B trial is at the same time as an occupational health intervention, and that will impose, in relation to a occupational health and safety obligation to monitor exposure at to a hazardous biological agent in the workplace. Now, because of that, we will have to reclassify COVID from being a class or group 4 occupational infectious disease to group 3 occupational disease, because now we have a sufficiently sensitive and proven intervention in terms of a vaccine, even though it comes through a phase 2, but phase 3A has actually given us that scientific basis to say we have a sensitive and effective intervention. However, ethically, we need to respect the informed consent in participation in the Sisonke trial. But legally, we need to understand that the employee is obligated beyond the Sisonke trial to participate in a vaccination program, as has been planned so far. There is also the WHO value of reciprocity. In other words, we will prioritize health workers and incentivize them in getting vaccinated earlier because they are more at risk. The relative risk of getting COVID, if you are a health worker as compared to you are not, is 2 to 3 times. And it is the combination of the ethics and the legal obligation and the special preference provided through the principle of reciprocity that I think are the things that need to be tied together so that we can be able to convince our employees to participate. And of course, the management of adverse events must be both as a clinical trial issue, as a quality management issue of a vaccination program, and as a COID issue. And lastly is that the governance of an occupational health and safety program through this infectious disease, the governance of a clinical trial, they create an opportunity of a balance between the ethics, the law, the occupational health and safety policies that have been issued by Department of Employment and Labor, DPSA and others, and program management, so that we are then having the obligation to collaborate across the different professions, but across different disciplines in terms of a transdisciplinary interaction. And this is the beauty of this work, that it imposes all of this. We can't, on a hard and fast rule, say, yes, it is obligatory and that is the only thing that we are going to be pushing. There's also the ethics of belonging or participating in a trial, and also the other issues that are supposed to be engaged with in relation to informed consent. Thank you.
Thank you very much. That was Dr. Sipho Senabe, the Chief Director for Human Resource Development and Employee Health and Wellness in the Gauteng Department of Health. And with not much further ado, just to inform you, we have brought Professor Elizabeth Maine forward in our sequence, and I'm going to immediately ask her to share her presentation slides after Dr. Senabe has unshared his current presentation. And by way of introduction, Professor Elizabeth Maine is Head of Academic Department Immunology and Associate Professor School of Pathology at the University of Witwatersrand. And she has her BMBSc, CH rather, at Masters in Medicine, and she's a fellow of the College of Pathologists. And there's quite a longer bit of introduction I can give, but I'm going to immediately hand over to Prof. Maine. Thank you, Prof. Maine, for taking the time.
Thanks, Ashraf, and thanks for the colleagues who preceded me. You've pretty much addressed most of my presentation, but I'm quickly going to give you just a little bit more of a basic science approach to the vaccine, and then at the end, what I'm going to do is I'm going to try and address some of the questions that have been asked in the chat, but also some of the questions that I think people have been asking me about the vaccine. Okay, so let's see if I can get this to work. So what is a vaccine? So a vaccine is any preparation which acts to induce or confer artificial immunity to a disease, and it stimulates the body to produce an immunological response to pathogens. And they've been called the greatest public health intervention of all time. And this is actually a picture of the last person to die of smallpox. And you might have heard that smallpox has actually been eradicated. So what do we want a vaccine to do? So there are three things that we can talk about. The first is control, and that's reduction of a disease in a defined geographical region. So if we're talking about COVID-19, we're talking about maybe the Southern African region or South Africa itself. Then there's elimination, and that is to stop transmission of the disease or reduce the rate of disease transmission to zero. And then finally, eradication means getting rid of the disease altogether so that we no longer need to vaccinate anymore, because that if infection for all intents and purposes no longer exists. How do we assess if a vaccine has worked? So there are three different things that you'll hear people talking about. The first is immunogenicity, and that just means, does the vaccine cause an immunological response? Then there's something called efficacy. So often as part of a clinical trial, we estimate how well the vaccine is protecting the participants from becoming infected or having illness with that pathogen. And then the third thing, which is obviously the one that we're most interested in, is effectiveness. And that's how well, if you were to look at the vaccine in the real world, where nothing's perfect, how well is that going to act later on? I think I'm going to go through this very quickly because Simba did mention it in his presentation, but we have four which are currently our major uses of vaccines. We either take a bit of something and use that as a subunit vaccine, for example, acellular pertussis. Then we have weakened pathogens that we can inject, and those are often called the live attenuated vaccines. And the big ones that we use are polio, the oral polio vaccine, and the BCG vaccine against TB. Then we have a killed whole organism. And then finally, we have this new sort of class of vaccines called the DNA and RNA vaccines. And this gives you nucleic acids, which will help you make the proteins associated with that pathogen. All right, now, this is a little bit of basic immunology, but remember that viruses, a lot of viruses are intracellular, or at least they spend most of their life cycle inside a cell. And so for viral infections, we have to kind of change the way that we look at immunity, because we're used to immunity being conferred predominantly by antibodies. But in the case of a virus, especially if somebody has the virus already, we need that viral protein to be sort of presented from the cell that is infected. Now, let's just look at how we develop immunity against the coronavirus. So here's coronavirus. It enters the body through the ACE2 receptor. It injects its own viral RNA into the cell, and it uses that RNA to make new viruses. Now, we have tried to see if we can replicate that infection process in terms of these vaccines. So the idea is, let us try and simulate, let us try and pretend that this is like natural infection. And really the only way for us to do that is to make sure that just like in normal infection, the DNA or the RNA from that virus is introduced into the cell, because that will stimulate the correct type of immune system response that will help protect us. Okay, so we have been looking at DNA and RNA vaccines. RNA is what is the genome of the novel coronavirus. And the RNA enters the cell and it uses the cellular machinery to make its own proteins, so it can make a whole new virus. And so the best way for us to simulate that kind of infection is for us to introduce RNA that's going to help make some of these proteins. And those proteins would then be picked up by immunological cells and used to make a response. Okay, now there's been a lot of concern about the safety of some of these agents. So mRNA vaccines, so messenger RNA, which is what we're using for a lot of the COVID-19 vaccines, has been tested, and this is not a new concept that we have. This is something that's been used for a number of conditions, not even all of them infectious, to try and stimulate the immune response and target it towards fighting against this intracellular bug. Okay, so here we have the viral RNA, and here we have a kind of delivery system. And in many cases, this is actually a virus, another virus, like adenovirus. And just to give you an indication, adenovirus is something that gives you like the common cold. That will then infect a target cell. That target cell will use that mRNA to make proteins from that vaccine, and will present them on the special molecule called MHC1. And this MHC class 1 will help to activate some very important cells of the T cell response, specifically CD4 positive T cells. And you might have heard about those in, for example, HIV, but those are kind of like the president of the immune system. So once they have become activated, they can then help the rest of the immune system to respond appropriately. Okay, and this introduction of these peptides is known as antigen presentation. And really, it's so important that that presentation happens in the correct way in order for the immune system to mount the correct response. So the DNA and RNA vaccines, as I say, are using the kind of correct way to stimulate the correct immune response against viral infection. Okay, so let's talk quickly about SARS-CoV-2 vaccines. Okay, so everybody is aware here that the Johnson & Johnson vaccine is being rolled out as part of a phase 3 clinical trial, and it's an adenovirus spike protein packaged vaccine. As Simba said, we're currently using a single dose, but there is a double dose trial as well, and it's stable at minus 20 degrees for up to 2 months. How effective is it? So this is something that we've been talking about quite a lot, and it's, you know, how effective is it against the novel coronaviruses? So again, it depends on what your aim is. And I think a lot of people are looking maybe at the aim being to prevent disease altogether. But one of the encouraging things about the clinical trials, and this is the paper that was published in JAMA, is that it protects you against death in about 100% of people. And even with the South African variant, we're thinking that there's a very strong protection against severe disease. Okay, then here are some of the other ones, and I'm just going to go through them very quickly because they're not currently here yet. We have the Moderna vaccine. We also have the Pfizer-BioNTech vaccine, the AstraZeneca, which is the one that we used previously but didn't show great efficacy, and the Sputnik vaccine, which was mentioned by Simba in his talk. Again, we've had a huge pipeline. We started off with 43. Only 7 have managed to go past the phase 3, with 3 approved, and the FDA has just approved the Johnson & Johnson vaccine itself. So how do they compare? So here are the different ways in which the vaccines can work. You'll see a lot of them are either mRNA vaccines, in which we actually inject the RNA directly, and then there's some which are being carried by a viral vector, like the Johnson & Johnson vaccine. Okay, so this is just a little bit of, background. Now I'm just going to talk about some of the very practical questions that people have, and some of your employees and friends and family might be asking these as well. So I got an email from a friend of mine who's not a doctor, and she said, DNA and RNA is our nucleic acids. Is this not going to change my genetic makeup? So this is a valid question, because obviously we're using a nucleic acid that is what exactly the virus uses. But this RNA vaccine does not actually enter your host genome. It doesn't actually enter into the part of the cell that codes for your own DNA and RNA. Some viruses, and these are typically retroviruses like HIV, do actually do this and can cause mutations in your own host cells. But the genetic material of SARS-CoV-2 is not inserted. So the virus itself and also the vaccine do not cause changes in your genetic makeup. What about the side effects? So I think that Simba and as we mentioned a lot of these, but a lot of these are fairly low grade side effects. So a feeling of not feeling well, maybe a headache, muscle pains, low grade fever, and generally they last for about 24 to 48 hours. There have been some very, very uncommon allergic responses up to the type of anaphylaxis, but nobody yet, or at least in the last study I read, nobody has yet died from anaphylaxis with the Johnson & Johnson vaccine. And it's approximately, as Azwi and Simba mentioned, about 5 per million participants. Just to mention that the side effects do seem to be more severe in people who previously had COVID, either the old or the new variant, and also seem to be more severe in people who get more than 1 dose. So in the 2-dose regimen, the second dose seems to be more associated with side effects. What about pregnancy? In the past, there was a theoretical risk, there is a theoretical risk of having this type of vaccine in pregnancy. However, the safety profile of these vaccines being what they are, and also the risk of contracting SARS-CoV-2 infection in pregnancy, has resulted in the WHO actually reversing its previous guidance. And now the recommendation is that women should be vaccinated at least in their 2nd and 3rd trimester for SARS-CoV-2. What about my medications? So a number of people have asked, I'm on corticosteroids, maybe I'm on antiretroviral therapy. What if I'm on pain medication? Won't they interfere? So a number of individuals may be on drugs which can suppress immune response, like, for example, corticosteroids and chemotherapy for cancer. There is currently no indication that this affects the efficacy of the vaccine, although we are continuing to monitor it. And as we mentioned previously, there's absolutely no reason you should not take an anti-inflammatory if you're suffering from severe side effects like injection site pain. All right, so what should you tell the person who is vaccinating you? So obviously you should mention if you're pregnant or breastfeeding. You should mention if you have a condition that affects the immune system like HIV or cancer or are taking any drugs. If you've had a previous allergic response, and in that case, we might need to monitor you for a little bit longer. If you're susceptible to bleeding, so if, for example, you have hemophilia, that's something that you need to let us know. If you have current symptoms of active SARS-CoV-2 or a fever, or if you've received another vaccine for SARS-CoV-2. A number of people have asked how long the vaccine protection will last. And we're currently not sure about that because we only have about 6 months of data on even the best vaccines. It may be necessary to have this vaccine more than once. And I see a number of people have asked about influenza vaccines and whether they should have the two together. Ideally, you should separate them. But again, it's always a good idea to take the influenza vaccine because it does provide protection for that year. The other question is, what about mixing and matching the vaccines? So in the UK, you might have read that they have actually utilized this approach. So given somebody, for example, one dose with the Pfizer vaccine and then followed it with the Moderna or another vaccine. There's been limited data on this, and we're not sure that it's completely efficacious. However, what we are starting to recognize now is even in the UK, which has obviously a circulating variant, which is quite similar to our South African 501v2 variant, there has been efficacy even with the first dose of the Pfizer vaccine. So that does provide us with some kind of comfort that this may be an approach which would work. What about in young children and what about in elderly patients? Now, again, there's been a lot in the public press about Europe where they have specifically suggested that some of the vaccines are contraindicated in elderly patients. Children under the age of 16 or in some cases under the age of 14 are not recommended for vaccination, and that is because they tend to be very unlikely to present with severe disease. So they get natural infection, but they don't become very sick, and they're much less likely to transmit, according to some of the studies as well. We have taken a principled decision that we are going to vaccinate elderly patients because elderly patients are at a risk of severe disease and death. So again, we're looking at our patients that do have high risk of comorbidities and severe disease, and we think that those patients should be offered the vaccine. What is herd immunity? And the president did mention this when he announced the vaccine trials. And that is what happens when a number of individuals, a critical number of individuals in a population are vaccinated. And they can then, because they can't transmit, because they're protected against contracting the illness, and even if they do contract the illness, will be much less likely to have a high viral load and transmit it. They protect people who, for whatever reason, cannot have the vaccine. All right, how do we know if the vaccine has worked? So there are different ways of monitoring vaccine efficacy. One of the ways that we're planning on doing this in NHLS is by monitoring people's antibody responses, and it will help show us whether the vaccine has in fact had the appropriate effect. All right, then another question that people have asked me quite a lot is, if I've already had COVID, I don't need the vaccine, I must be protected. Now, one of the things we know about coronavirus species in general, and SARS-CoV-2 in particular, is that it is possible to get reinfected with the disease. So in some patients, infection does not confer full immunity. In addition, coronavirus mutate, and so there are a number of variants which are circulating in South Africa, which may result in reinfection. The important thing to remember here is even if you do acquire coronavirus after you've been vaccinated, and we know that there's not 100% protection, even if you do contract it, all of the data suggest that you will not have severe disease. So again, we need to do some expectation management to say you might still get coronavirus, but the bottom line is you're probably going to be protected against severe disease and death. Okay, so I saw that the previous speaker spoke about ethics and the Occupational Health Act, and I'm not an expert in public health medicine, but certainly discrimination against individuals who have not been vaccinated is not currently something which is supported. I think there may be changes in that, but currently you cannot force anybody to have the vaccine. All right, so those were some of the questions that people were asking. I don't know if there's other. Oh, so I've got a couple of questions. I don't know, Ashraf, do you want me to just go through the?
Yes, please, Prof. Maine, please proceed. And the question from a colleague about pregnancy and breastfeeding, which vaccine, or is there still more progress to be made? Thanks, please proceed.
Okay, so as I said, currently pregnancy and breastfeeding is not a contraindication to any of the approved vaccines. Previously, there was a question over that, but the WHO, as I said, have reversed its guidance on pregnancy and breastfeeding. I think it is important for the vaccinee, the person being vaccinated, to disclose that they're pregnant and or breastfeeding, but it's not a contraindication. Some other questions that I've got. I would like to know, if one takes the vaccine, what is the vaccine's lifespan? As I said, I'm not sure, and I think it's probably going to be the case that we might need to have a yearly vaccine. It may be the case. All right. Ivermectin. So ivermectin is an anti-parasitic agent, and currently, I think a lot of people are using it for off-label use for SARS-CoV-2 as an immunomodulatory agent. Because it's currently only available on compassionate grounds and can only be used on a named patient basis, it hasn't really been studied in terms of what contraindications it might have for the vaccine. It will be something interesting for us to watch. So there have been a number of cultural caveats to the J&J vaccine, and one of the issues that has been raised by some religious leaders is the use of fetal cells. It is halal for those individuals who are Muslim, but this is something that I think we're going to have to engage with the religious leaders for. Can a person receive the hepatitis B vaccine as well as the COVID vaccine? Yes, you can have both vaccines. You should try to separate them. So try not to have both simultaneously, as they can interfere. And there is a suggestion, although I don't have clear data to show this, that there might be a more severe immunological response in terms of side effects. Can the vaccine be given to people with penicillin allergies? For sure, it doesn't contain any penicillin. Should vulnerable people be included as vaccinators? It depends what you mean by vulnerable. Have I been vaccinated? No. I'm hoping that I'm going to be vaccinated in the next few days. And I think is the principal investigator going to receive an honorarium? I think that this is highly unlikely. I'm not sure, but I'm not the right person to answer that. How would the risk, what about the test after COVID-19? It will not have any effect on the PCR test result. If you're infected, you'll test positive with PCR. If you've just had the vaccine, you will not test positive for PCR. I think that's all the questions that I can see, Ashraf. I don't know if I missed anything.
Thank you very much, Prof. Your time, I know, is under pressure, and for making the time, I thank you very much for joining us today. And we hope that we can rely on you to join us for, I can see the necessary follow-up sessions and webinars that's going to happen. Thank you, Professor Elizabeth Maine.
Thanks, Ashraf.
And Professor Maine is the head of Department of Immunology at Charlotte Maxeke, Johannesburg Academic Hospital. I now immediately am going to hand over to our other presenter, that's Dr. Lumka Puwani, who has kindly swapped his position with Prof. Maine, given that she had certain time constraints and pressures. Dr. Puwani is an occupational, he will be dealing with an occupational health by clinical trial in vaccination, and he's an active in-house occupational medical practitioner and will provide us with that particular perspective from the Steve Biko Academic Hospital. I saw that you had tested your presentation slides and they were working and in full screen. If you could do so again and just check your audio for us, Dr. Puwani. And for everybody who is with us, just a reminder that we will be sharing all the presentations and the slides, and this full online video recording of this particular webinar will be available on our YouTube channel as well. Please visit the NIH's website with a huge number of very useful resources for us to ensure that we strengthen the COVID-19 prevention and outbreak response capacity within all of our workplaces, given the many waves that we're facing, as well as the now development and unfolding of the vaccine programs. So please visit the www.nioh.ac.za. That's www.nih.ac.za. You can email any follow-up questions you have that may or may not be covered by Dr. Puwani in his presentation to that is info at nih.ac.za. That is info@nih.ac.za. And then we also have the red cap. We must ask you for your feedback on this particular session, as well as your suggestions for any other and important and critical topics that you feel that the NIH's COVID-19 webinar series must cover. And therefore the red cap survey that I will again post in our chat box is where you need to go. Please just copy the link from the chat box and do the post webinar survey for us. We in particular want to know from you what are the topics you're interested in having. So just checking up again with Dr. Lumka Puwani. Dr. Puwani, are you with us? I can see that your microphone is open. Can you share your slides? Otherwise, I can ask our IT colleagues if they could do that for you. Okay, so we've had quite a bumper number of questions. We've had over 130 questions covered, besides the very detailed list that also covered by Professor Maine. It's important if Prof. Maine could just also just share that presentation, or we have it and we can share it with the colleagues. I just misread a note from a colleague. So just to follow up again, I'm going to ask our IT colleagues just to double check on Dr. Puwani's connection. I can see a microphone, but not a camera icon for Lumka Puwani. And maybe if I could just check with him on his audio. If his audio is okay, then we can share the slides from here. Dr. Puwani, are you there? Okay, so that's slides being shared. If it could be maximized. I'm not sure if Dr. Puwani has microphone problems on his side. Okay, so a reminder then, The Q&A box is where your questions go and your general comments in the chat box. We would like you to do the survey post webinar, and I will be posting that link. Visit the website. We also have a Twitter, I think it's called a channel, where quite a lot of information is shared there. But also, in addition to the website, we have the YouTube channel where all of our webinars and the full recordings thereof is made available. Okay, so I'm just going to require somebody to check with Dr. Puwani about his sound. I know he is in an active clinic at the moment, and he's joining us from that very busy clinic, and it seems like we might have a bit of technical problems there. So let me just quickly share again that link for you. It's important that you provide us with the feedback so that we could ensure the topics are covered and any other comments you might want to provide us to improve our NIH webinar service. So the particular link I'm posting now in the chat box, and I'm just going to check with a colleague if they could contact Dr. Puwani to hear from him if he has any particular difficulties on his side. So I've just posted in the chat box the link to the red cap link. If you could just click on your chat icon at the bottom of your screen, there's a little chat bubble there. Not the double bubble, that's for the Q&A, the single bubble, and you'll see that I have clicked for you, posted for you there, the online link to the red cap for the NIH COVID-19 webinars feedback survey. So, okay, we are attempting to get hold of Dr. Puwani. He's a practical experience in the clinical setting, and his previous contributions to our webinars has been invaluable. And I would definitely love Dr. Puwani to be part of this webinar, and we will try our best just in the next minute or two to get him on board. And then, so the link I've given you for the short survey, which maybe some of you could open up separate from the Zoom session and click on the https redcap.link NIH COVID-19 webinars feedback survey. We will be posting this after each of the webinars we hold. For those who had asked questions about certification, we have the general certificate of attendance, and that we circulated to all of those who have successfully registered on the attendance records of the Zoom webinar. For those who require the CPD link for the CPD certification, we will send out the CPD link to the test as well as the password after the webinar once we've completed the uploading of the CPD questions. For those who do belong to professional bodies that have accredited this particular webinar with CPD points, not all bodies are occupational health specific and related. So you will have to use that link on the ClassMonker platform to do your CPD test. And then in terms of that particular test, once you've completed it successfully, it requires a minimum of 7 out of the 10 questions to be answered correctly in order for you to be able to complete the test and then get the CPD certificate for that. That will be circulated within 24 to 48 hours after the webinar, once we've completed the capturing of the particular set of questions and done the formatting and design of that test. I'm going to ask my colleague Tabani just to double check with Dr. Puwani about his availability. He's definitely logged in on our Zoom session. I can see that his microphone function is activated, however his video function is not. So he may or may not be able to see that we have actually shared the presentation slide on his behalf from our IT colleague side. So let's just double check on that. Otherwise, we may have to incorporate Dr. Puwani's presentation in a follow-up session on the vaccine. We have had more than 1,000 people logging in, and we only have a maximum capacity of 1,000 Zoom license for logins. We definitely had way more than 1,000 interested attendees having registered on our session. And this particular session has been so popular that besides the questions answered by Professor Elizabeth Maine, the head of Department Immunology at the Joburg Gen, there's been over almost 140 questions being asked in the Q&A box and answered by our capable group of panelists. Dr. Puwani, are you there?
Hi, Ashraf.
Hi.
Can you hear me now?
Yes, please proceed.
Oh, thank you. Sorry, had experienced technical glitches. I could load the slides, but the audio was the issue.
Yes,
but
please
okay.
proceed, no problem. I think your presentation is still important as part of our overall program design. Thank you very much for joining us and making the time from your very busy schedule. Thanks. Please proceed.
Thank you so much, Ashraf. I'm going to quickly share with you our experience at Steve Biko from OHS perspective, occupational health and based on this clinical trial or on the rollout of this J&J vaccination rollout plan. Just to give you a brief background, as we all know what led to the sequence of events that made us to be where we are now. With the second wave of COVID-19 came hope in the form of COVID-19 vaccines. And the developed world had had an early start, and South Africa had just received its first batch of vaccines, which was AstraZeneca then on the 1st of February from India. And Steve Biko, as one of the accredited site, was in line then of receiving a portion of vaccines on the 15th of February. And this then afforded us time as occupational health practitioners and the whole staff from occupational health to workshop and educate our staff on the COVID-19 vaccines. But then we can move to the next slide. As we all know by now, the reasons why we then moved to the J&J vaccine. It was because the AstraZeneca failed the efficacy, especially in covering the strain, the predominant strain that we have in South Africa. So as the initial presenters have presented to us, that we all know that there's a phase 3B clinical trial of Johnson & Johnson COVID-19 vaccine, and selected centers, which Steve Biko is one of them, have been accredited to roll out the J&J vaccine in the past 2 weeks. And this study involved 100,000 participants. And of these participants, it included the healthcare workers, both public and private sector. We can move to the next slide. And then our experience on our side from Steve Biko in preparing for this rollout. I'm just going to mention to you the most important parts or collaboration or elements of collaboration which were important to us to ensure that the rollout is a success. The clinical trial in our setting was led and conducted by research groups such as the Medical Research Council, Synexus Research Group, and Sichaba. And the study name, as we all know, is the Sisonke, and the study population comprised of healthcare workers. The next slide. So in our setting, it was very important to have collaboration between the Department of Health, the research groups, and occupational health. As we all know that occupational health played an important or an integral part in ensuring that all of our healthcare workers get to be vaccinated. The next slide. And in order for us to ensure that all the staff members or healthcare workers get to be vaccinated, we had to develop a risk-based classification, which was primarily based on the risk of exposure to COVID for our healthcare workers. So in our classification, we had category 1 up to category 4. And category 1 was basically the staff members that are working in or with COVID-19 patients. That is our staff members that are in the emergency unit and in COVID wards and in COVID theaters and ICUs. So those ones, we then classified them as category 1, and of which they were going to be the first ones to be vaccinated. And then category 2, we had OHS staff and infection control unit and all of our doctors, that is all the staff members that are patient facing, all our allied workers, waste collectors, and those that are working with their linen. And with category 3, we had pharmacy, admin clerks, household staff, cleaning units, porters, drivers, and the engineering staff, security and food services. And then the last group, which is category 4, was those that are not treating or facing patients. That is category 4. And then we can move to the next slide. So of importance then in the preparation, the elements of collaborations were mainly those 5. That is communication and good planning, and with resource allocation, data collection, and the vaccination rollout as a whole. We can move to the next slide. So when it comes to communication, we had regular webinars and team meetings which were held before the onset of the research, involving all the relevant stakeholders, that is the Department of Health and our CEO, and with occupational health and IT, communication department, pharmacy, admin and logistics, and security. The Medical Research Council was also part of these discussions with the Sisonke group and the Synexus group. The next slide. So as occupational health team, we played a very important role in educating and training our healthcare workers and encouraging them to enroll on the EVDS system, which is our electronic vaccine data system for the rollout program. And all stakeholders involved presented to the managers, hospital staff, and to organized labor. And non-verbal communication aids such as the signage were developed and posted, and SMSs were sent to all staff members who have registered and those who were encouraged to register for the vaccination. And then as soon as the vaccination rollout occurred, we had meetings, debriefing sessions every day in the morning and in the afternoons. We can move to the next slide. And then when it came to planning, one, we had to identify a site that fits all the infectious control measures, and a site that could be accessible to all of our healthcare workers. So we managed to identify the site and was prepared for the rollout. And of importance, the issue of consent forms was then developed by the research groups, which was then distributed to all of our healthcare workers that have registered for the vaccination. And we also had the adverse event reporting tool in place in case of all the allergic reactions or the adverse reactions that were experienced and then reported. And then for us to avoid queues and backlogs at our vaccination site, we then developed an appointment system, a pre-booking system, which was through EVDS and the Sisonke group, and also from occupational health. So we were linking all these pre-bookings via occupational health in order for us to allocate our staff members with different slots, because the other thing that we wanted to avoid is delays or disruptions in service delivery. So we had that system that each and every healthcare worker that is booked on a certain day is expected to present himself or herself at a specified period of time, so that we avoid a long waiting period at the vaccination site. And then the investigation product, that is our vaccine, it was handled basically by the Synexus group, their pharmacies, and they brought it to the site every morning, every day, received from the storage facility from Synexus in Watermere, and distributed to the vaccination site on a daily basis. Security was an integral part or an important part on handling of the vaccine, that is our IP. And then information and technology was also important, as they provided all the necessary IT preparations for the rollout. And all of our vaccinated got time to be trained and be educated on the whole process of vaccination. And infection control measures such as waste handling, etc., were also dealt with accordingly. And we can move to the next slide, which is resource allocation. So human resources in the form of vaccinators and runners was organized. Data capturers who were able to capture all the data necessary for the whole program. And for that, we needed computers, laptops, tablets, data and routers, telephones, and of which they were provided from supply chain and National Department of Health and IT. Fridges and cooler boxes for pharmacy for the storage of the IP were also provided. Photocopying machines and sanitizers, they were all provided in our human resource allocation. And then when it came to data collection, data, which is the mainstay of research, was collected at real time on site by data capturers from both Steve Biko group and research group. And data on the first 2 days, it was captured on paper and on computers. But on day 3, once the rollout occurred, we went paperless. And data collection included identification of healthcare workers, the recording of the IP doses, the batch number, and recording of the vaccinator's name and the number of workers vaccinated. Then on the vaccination side, we had our queue marshals who were there to manage our queues and also to direct our healthcare professionals as to where they're supposed to go. And the vaccinating team, they were all trained and led by occupational health practitioner and staff, and vaccinated the healthcare workers. So this was basically our plan for the plan and the executing of the vaccination. But the issues of concern that then arose once the rollout was on was the conflict management during the operation was not prepared for adequately. And there were booking challenges experienced, especially in the initial stages by our healthcare workers. And then the third issue was the demand and the large numbers of healthcare workers who came on site for the vaccination, and at times which led to the IP shortage. The larger numbers also put a strain on infectious control measures. And the fourth issue was the security issues where at some point the South African police had to be called to come and assist the queues and the large number of people that were in our premises, all coming in for vaccine, and long working hours, which also included weekends. And then the last issue was the training, where in the beginning, most of our vaccinators were not properly trained, and then they required more of assistance in that regard. I think, in short, that's about all on our experiences as occupational health in this rollout of the J&J clinical trial.
Thank you very much. That was Dr. Lumka Puwani, an occupational medical practitioner at Steve Biko Academic Hospital, and I had confused you with a male colleague here locally at Charlotte. My apologies to you. Thank you very much for that amazing contribution. And at this point, I need to just thank all of our presenters, starting with you, Dr. Lumka Puwani, the occupational medical practitioner at Steve Biko Academic Hospital, one of the key deliverers of the vaccine rollout as a center within the Gauteng and Johannesburg, Gauteng area. Thank you for your contributions. And then also to our other presenters, Dr. Simba Tavuka, Dr. Azwi Takalani, both are medical monitors with Sisonke Open Trial, and then they were followed by Dr. Sipho Senabe, the Chief Director, Human Resources Development and Employee Health and Wellness from the Gauteng Department of Health, and Professor Elizabeth Maine, who is a colleague in the broader NHLS, of which the NIH is a part, a specialized division dealing with occupational health. Professor Elizabeth Maine in the NHLS is the HOD for immunology at Charlotte Maxeke Johannesburg Academic Hospital. Thank you very much to all of our presenters for joining us today, and then also for the number of panelists that has contributed to our session today. I just quickly want to get to their list of names. And if I could just quickly scroll here, I will definitely find them. The list of panelists that have contributed today is Dr. Simba Tavuka, obviously, Dr. Azwi Takalani, Dr. Senabe, Dr. Puwani, and then from our internal colleagues, Dr. Odette Volmink, Dr. Mazvita Mberi, Dr. Hlonipile Marshall, Dr. Edward Sepirwa, Dr. Nkululeko Tunzi, Dr. Edzani Mpapuli, Dr. Nompumulelo Ndaba, and Dr. Tendo Mpapele. And in particular to my two colleagues in the IT department, and that's Glenn, as well as Tabani, that is Tabani Zwane and Glenn Vongani Mashele. You've played an amazing role today with quite a number of technical curveballs we have received. And on that note, I'm going to again remind you to please do the REDCap survey to all of those who've attended. I will just briefly again post that particular link for you. It is an important new initiative that we are undertaking to ensure that we are getting maximum feedback from you to give input to our planning of our online COVID-19 webinar series. So I'm just quickly posting again that link for you. It's important for you to, as soon as you're done with the particular webinar, to visit that particular link and just do the survey for us. I will also post, switching off my microphone, play that set of slides that will give you some key information with regard to the post-webinar administration. And on that note, thank you for joining us here today at the National Institute for Occupational Health's COVID-19 Vaccine in the Workplace and Overview webinar. we will see you on our next webinar. We've got a critical webinar happening and focusing on health and safety representatives as a particular target audience. That particular invite, I think, has already been circulated, and we're encouraging all of you to make it possible for the health and safety representatives, the SHE representatives, particularly those in the public sector and government departments, to please join that webinar coming up. I think it is on the 9th. And with that, let me just quickly share the particular slideshow. It's this one over here for you to see the important, oh, now I need to get to this one over here, the important information. And I've started that for you, and I'm sharing it